Sorry. Didn't know I was going to cough, or I wouldn't have started the thing. So we're going to talk about some of the mechanisms of epigenetics. A lot of times what you've got is the attachment of a methyl group, not usually in the gene itself, but to the promoters, the recognition molecules that you need to get to in order to get to the gene. Very typically methylation of cytosine, so it does actually attach to the DNA, but it doesn't change the sequence. It just makes recognition not work. You can't recognize the sequence if there's a methyl group in the way. So it's a basic similar idea to direct enzyme inhibition, but of course this is not enzymes. But the idea of getting something in the way so that you can't attach is basically the same thing. Now there are some times where having a methyl group actually increases the ability to recognize and express the gene. Remember, epigenetics is primarily gene expression. Is it, can you use the gene, can you not use the gene, can we activate the gene, can we deactivate the gene? That is most of what is happening in epigenetics. If the methylation is stripped off, the gene activates. This is sometimes called imprinting, where through the course of your lifetime, you clip stuff onto the chromosomes, and then when you make copies of the chromosomes, you leave that, you duplicate the clip as well. So as cells get produced, they're equally deactivated. Typically, when producing sperm and eggs, one of the things that happens is most of the methylation is stripped off, but some of it is not. And imprinting is very often applied to what's still under kind of lock and key in a sperm or an egg cell. Some of the matched genes, the genes that both the male and the female contribute, are activated in the egg cell and deactivated in the sperm cell. So early on during embryonic development, there are genes that are contributed by both the mother and the father. They're on chromosomes with in a homologous pair, but only the mothers are actually being used. And then later, and then when the X chromosome is shut down, remember Barr bodies? One of the X chromosomes is shut down to make X expression equal in males and females. There's a lot of imprinting of both methylation and a lot of coiling. Very similar coiling to what happens when cells are getting coiled up, or chromosomes are getting coiled up in prophase. Acetyl groups can be attached to histones, also very commonly used to fiddle with proteins. And of course, if you can clump shut an area of DNA with genes on it, then you can't get at the genes. So in some cases, epigenetics doesn't go after the gene or the DNA near the gene, it goes after the wrappings, so that you cannot unwrap it. There's a term I ran into in the last year. Remember, histones plus DNA are chromatin. And there's a term called active chromatin and inactive chromatin. Inactive chromatin has been acetylated in a way where you can't unwind it. Sometimes temporarily, sometimes permanently. When we talked about DNA and RNA, I mentioned that RNAs has been a real big growth industry in the last 15 years or so, when people discovered that there were a whole bunch of little RNAs in cells, mostly in the nucleus, that did all sorts of things. And they were being produced from DNA codes, but they were not messenger RNA, they were not transfer RNA, they were not the RNA everybody learned in basic biology. They were little strips of RNA. At the beginning, they were called microRNAs. And they interact in the nucleus to do various things. And just to give you an idea of just how crazy this gets, I have a list that I'm going to be copying off of. You're going to want to leave yourself room here. It's gotten so, I'll pick up an article and I'll run into one of these abbreviations, and it's like, fortunately, before very long, microRNAs was just microRNAs, and you figured you knew what you were talking about. But then they started to pin down what different ones were doing and started to change the prefixes here. And for people who worked in the area, they knew what they were. Most of the time, they would tell you what they stand for. Sometimes they don't. And I've got to look at it myself. The LNC are long non-coding RNAs. They're small non-coding RNAs. So it's not long and short, it's long and small. The piRNAs are Pee-wee interacting. It turns out that there's a class of proteins active in the nucleus called Pee-wee proteins, PIWI. SI are small interfering RNAs. They get in the way of stuff. And MI and microRNAs are just kind of a catch-all for little RNAs that they probably will eventually give more initials to as they figure out what they do. But this gives you an idea just how crazy this area is. There's so much work being done in it. And there's always a list of human conditions and diseases on the list of we don't know where the heck these things come from. And as they discover the functions of these, occasionally things get checked off the list. Oh, this is associated with Pee-wee proteins, and the Pee-wee RNAs are not functional properly. It is possible to get epigenetic mutations, but that gets really confusing. Mutations are generally considered to be changes in the DNA. So if you get changes, epigenetic changes that change, the important stuff, you will sometimes see them called epigenetic mutations. I'm hoping they will back away from that and stop calling them that and come up with, a general term for epigenetic changes that are inheritable. And, will change both this cell and the cells that you make from it will change both, will be in this organism and passed on to the offspring of the organism. But it's still quite the state of flux going on.